Okay, so for the next 5 or 10 minutes, I'm going to talk about some enhancements of our ibody platform and also some additional activities in regards to product development for AD214, specifically from a process development perspective and a formulation perspective. So I'm going to start with the ibody platform initiatives. We all know that we have a great platform. We've been able to isolate specific ibody binders to at least 20 targets, so it's a great platform. But we're not going to rest our laurels there. We want to make a great platform even better. So we've identified some focus areas which we're going to be looking at in the near term. Initially, discovery speed, so I'm talking about high throughput, how quick we can get projects through our pipeline. And secondly, we're going to be looking at improvements from a manufacturing perspective. So what I mean there is implementing some initiatives such that the ibodies that we get out of our screen are generally more manufacturable. What I mean by manufacturable, I mean high expression levels and stability and solubility. We're hoping the initiatives to focus on those two areas I just discussed will lead to further IP extensions. And ideally, we'd like to implement those in the short term. Excuse me. Moving to the product development aspects. First of all, from a bioprocess perspective, we obviously have an appropriate phase-appropriate bioprocess as demonstrated by our recent successful manufacture of our drug product for our phase 1 clinical trial. Everything went to plan there. There was nothing unexpected. However, like all biologics at this early stage of development, there is opportunities for improvements. It's a work in progress, and certainly we're going to be looking at improvements in yield. And that's just not from a commercial viability perspective. Obviously that's important, but enhancement and yield are going to give us a lot of flexibility in regards to the type of indications we can look at, the routes of administration, and also the dosing schemes. As with any major change though, the earlier you can bring that into the process, the better from a risk, a regulatory perspective and a comparability perspective. So we're aiming to introduce changes before phase 2. Moving on to formulation enhancements. So at the moment we have IV delivery. The formulation is obviously appropriate for IV delivery for IPF. However, at one point we might want to look at pulmonary delivery. Obviously that would be a complete different formulation. We might want to look at the optionality of 214. It's involved in a lot of fibrotic conditions, as Mick mentioned. A lot of those would require different forms of delivery. Macular degeneration would require an intravitreal delivery. Obviously, once again, that would be a different formulation than what we currently have. We're looking at formulations which will improve patient and clinician convenience. At the moment, it's IV, subcutaneous, as was mentioned by the gentleman previously. It's much more convenient from a dosing perspective, so we look at formulations along that road, and additionally shelf life extension. So at the moment our drug product is stored at -80 degrees. That's not particularly convenient. Certainly we'd like to have formulations which are stable under refrigeration conditions, and also look at lyophilization as well. So there are a lot of formulation initiatives which can be taken to try and open up the optionality of AD214. After the phase 1 study, when we get a bit of an idea of the dosing schedule, that will certainly inform the type of formulation studies we'd like to do. So focusing initially on the ibody platform. On the left, this is just a list of where we are today. On the right, this is some improvements we'd like to make in the library. As of today, our core patent expires in 2025. Obviously, we like to get extensions to that. Certainly, we want extensions above and beyond just an IP land grab for individual targets. That's easy, we will do that. However, we're much more ambitious than that. We really want to implement changes to the library which will extend the protection on the actual platform itself. Where's that going to come from? If we look at our library today, our scaffold, it's obviously very robust at the moment. However, looking at structural data and sequence data, you can see that there are certain residues in the framework which could be modified, which may lead to improved manufacturability, also may lead to improved stability of the ibodies that we isolate from the library. And this, we hope, will directly address the patent protection milestone. The second goal for the ibody 2 platform is to improve the discovery efficiency. So at the moment, our throughput is certainly adequate for our in-house programs and also the 1GE program that we have at the moment. However, as well as planning for success, we're actually expecting success. So to be able to take in more programs, we need to improve our discovery efficiency from a high throughput perspective. You look at our current library, there are certain initiatives which can be undertaken which would enable a high throughput process to be implemented. We will implement those in any future library construction process, particularly those process steps which we can eliminate. Additionally, we'd like to increase the amount of automation that's in the process. That certainly increase the throughput. And additionally, we'd like to bring in some technologies which allow us to bring some activities which are generally done later in the process much more forward into the process, like looking at binding to whole cells. So that certainly addresses this issue here. Improve pharmaceutical properties of ibodies. It's common knowledge in the bioprocessing industry that FC proteins are much more difficult to manufacture than antibodies. If you look at our current processes, at the moment when we construct our library, we don't really have any selection processes which are going to favour those sequences which are generally going to be more manufacturable. So we've identified some of those processes and we're going to integrate those into the construction of future libraries. End result of that is we're going to decrease both the cost and the time for product development and manufacturing development. So moving away from the ibody platform onto continuous improvement from the downstream and upstream perspective. As I said, we have a phase appropriate bioprocess at the moment. As I said, our manufacturing run went as expected. Looking at our GMP process, to be quite honest, the downstream process, it's really solid. But we can certainly see aspects of the upstream process where there's clear room for improvement. And we'll be undertaking studies to see if we can realise some of those upstream improvements. Yield's incredibly important in the bioprocess industry, particularly if you look at these two following points here. At the moment, it can be quite difficult to access contract manufacturing organisation facilities. There's only a certain number of them around the world. They only have certain capacities. Once you go over 2,000 litres, you start to move up into the stainless steel tanks, the larger 5,000, 10,000 litre tanks. They're much more difficult to access. So the higher you can get your yield, the lower capacity you need to access. So that's certainly one aspect of yield which is going to have a big impact as we move forward. Obviously, yield's going to impact the cost of goods. At the moment, our current COGS may cause some issues in regards to the maximum dose that we can do, the route of administration, also some indications. So by increasing our yield, this will release some of the pressure that we may have further down the track. Looking at formulation developments, as I said, we have IV administration at the moment. Certainly not the patient's preferred mode of administration. Subcutaneous, much easier. Bit more of a challenge from a formulation perspective. There's a bioavailability issue. There's also a protein concentration issue. Generally, subcutaneous deliveries are well above 100 mg per mil or at 25 mg per mil. So there's certainly some formulation activities which would need to be advanced there if we were to be able to move into a subcutaneous administration regime. So why do we need to do those now? In biologics, changes take a long time. So it takes a fair amount of time just to identify the change that you want to make. And then once you've identified that change and you've shown that it works, you then need to do comparability studies. So you need to show that the product you make with your new process is the same or extremely similar to the product you made with your previous process. So because of that, it takes time. We'd like to get it all done before phase 2. For these reasons here, we want to make sure that we can look at higher doses and we can look at different forms of administration. We certainly want to improve the clinical convenience. We want to minimise our requirement for large CMO capacity. And obviously the biggie here, it's all about the dollars. We want to decrease the cost of the product. We do have plans in place. They're quite detailed. They're with our manufacturer KBI, and we hope to be able to investigate those in the not too distant future. So this is really just a summary slide. So you look at the ibody 2.0. We're going to evaluate alternative scaffold sequences. We already know what changes we want to make. We know how we're going to measure them, so that should go quite smoothly. We're going to integrate the high throughput enhancements that I spoke about, and also look at the selection processes, which hopefully will provide higher quality ibodies after the screening processes. We're going to then synthesize these libraries, QC them, and make sure that the processes that we've introduced do actually have a positive impact. And then hopefully it will lead to new IP, which we'll file. That's going to be about a 4 to 6 month process. So in the terms of biologic development, that's actually not long at all. Looking at our product development issues split out into the process development, as I said, it's mainly the upstream yield we're expected to get pretty good gains from. We've got detailed laboratory scale plans with KBI to address yields. It's about a 4 to 6 month program with them. Once you do find conditions where you do increase your yield, you want to move it to manufacturing. At the moment, there's about a 12-month wait period to get into any manufacturing facility. Moving over to the formulation, well, this is going to be indication specific. We're going to be looking at formulations which enhance the shelf life and also even the in-use life of the product. And we're going to be looking for formulations which will allow optionality in regards to different forms of delivery for different indications.